So we're going to discuss, generally speaking, proteins and what they do, focusing in particular on their role as biological catalysts, i.e. enzymes. So in terms of biological molecules that are found in an animal or plant is not that straightforward, but found in animal body, virtually all the functions from structural to signaling to catalytic are carried out by enzymes. Enzymes are formed as polymers from 20 different amino acids. Now, full disclosure, currently it is accepted that there are 21 amino acid because there is an amino acid called selenocysteine. I will illustrate it. The function, the structure and therefore the function of proteins is determined by amino acids that comprise them, not only the kind of amino acids, but also the order. The functions of proteins can, hair, a big chunk of bone, cartilage, muscle. Structurally, we are, for the most part, proteins. Another really important function of proteins is enzymes, biological catalysts that can speed up certain biochemical reactions. They can transport gases and store them. Myoglobin stores oxygen, hemoglobin transports it. They signal. They protect. For instance, a lot of protective functions of innate and adaptive immune system are carried out by proteins. Okay? Because of them, we can move. But before we get to the polymers, we need to chat about the monomers, the amino acids. So all amino acids share the same structural motif. So they have a central carbon atom called alpha carbon. This alpha carbon is connected by two functional groups that should be familiar to you. The ammonium group on the left and the carboxyl group on the right. This central atom, this alpha carbon, forms two other bonds, one with hydrogen and one with the side chain, also known as functional group. So before we move on, what just happened? I don't know. So before we move on, here's what I want to emphasize. All amino acids, with one tiny little exception, actually all of them, have the carboxylate group, the alpha carbon, and the ammonium group arranged in that way. And all of them differ because of the R group. So, since the only part of an amino acid that tells it apart from other amino acids is a side chain, amino acids are classified based on the polarity of their side chains. If the side chain is, let's say, aliphatic, basically a hydrocarbon that's attached to the rest of the molecule, is nonpolar. This is an example of a valent. If the side chain contains organic substituents, functional groups that make it polar, for example, carbonyl group and amino group both make this carbon fairly positively charged, we call it the polar amino acid. Within each group, there is a bit of a division, okay, which we're going to discuss. And this is your first set of non-polar amino acids. You probably remember from our previous conversations that the bond between the carbon and the hydrogen here, and between carbon and carbon, those bonds are non-polar. So glycine, alanine, valine, leucine are all, and so on, all are non-polar amino acids. Okay? So yellow boxes here highlight the side chains. Now these amino acids are sometimes called aliphatic non-polar, and these are often called aromatic non-polar. Okay? Proline is a cyclic one. It's slightly different. Technically, it's also aliphatic. You see, the amino group looks slightly, different one. And methionine, shown here, is also a fairly non-polar amino acid because this sulfur does not really provide much of a polarity to the bonds. So when we're going to cover different types, I will explain what you can expect on your fourth and the final exam. Now, polar amino acids can be charged or not. These are polar neutral amino acids, which means they side chains carry no charge. From serine to glutamine. If you would look at each amino acid, you would see that there is a polar bond between carbon and oxygen here, carbon and oxygen here, carbon and oxygen here, and these amide groups are really, really polar. But there is no charge, as you can see, right? Right? Makes sense? Now, this is cysteine. Remember, I told you there is a 21st amino acid? Not for the exam, just FYI. So instead of SH, it's going to be selenium H at the end. Okay? Does it make sense? Can you take a look on the periodic table for me? Where is sulfur? Yes, number 16. And selenium? Right under it. And what do we say about the elements in the same column? What do they have fairly in common? Broadly speaking.
The electrons on the out.
Which means, what do they have fairly common?
Same charge.
Broader. The
Electronegativity.
P word.
Or
Proton.
Raw properties. They have similar chemical properties, right? Does that make sense? Remember, folks, if I would tell you, look at the group 1, you would tell me this right away. I just didn't want to make it easier for you. Because you remember, all those are alkaline metals. They all act the same, you see? So remember, columns, similar chemical properties. So it turns out, as we will see, this group is a very good donor of electrons. And selenocysteine is found in a lot of proteins that deal with so-called oxidative stress. So now let's take a look at amino acids that are charged. So we have two amino acids that effectively have another carboxylic group, in the side chain. At the neutral pH, the physiological pH, this group becomes deprotonated. So hydrogen that was supposed to be here isn't. At physiological pH, they dissociate. Does that make sense? Which makes these two amino acids negatively charged.
So I have a question.
Sure.
Like in the first example of an amino acid that we were shown, it looked like it was structured. Like it was like the carboxylate group was like
on the side.
On the side, like so, would the alpha carbon be for those two, for example? Would it? It would be that.
Yes.
So what is below that functional group there?
Which one?
The top, very top one. So like the CH2, would that be the?
That's the part of the functional, that's the part of a side chain. So everything that is within the confines of a white box is a side chain. Side chains can have different it may not have any functional groups. This side chain has no functional groups, right? Just 3 carbons with some hydrogens on them. Fairly non-polar, nothing to see here, not super chemically reactive. Does that make sense?
Yeah, so I guess, and also like where it says an amino acid is non-polar when the R group is H, alkyl or aromatic. So how do you always identify, how do you always know what the R group is when you see like that?
When you see a random amino acid, that's what you're asking.
Yeah, because like in the first example, the R group was methyl. Like in the...
Right.
Yeah.
So how do you identify it? Here it is. You should look at the amino group, ammonium group, hold on a second, and the carboxylate group that are linked by a carbon.
The alpha carbon.
That linking carbon is an alpha carbon. No matter how I draw the amino acid, an amino and the carboxylate group will be connected by the alpha carbon. I'm going to pause right there and ask, does that make sense?
Yeah. And the alpha carbon will always just be carbon. Would it ever be like CH?
Well, technically there is a CH here, right?
Yeah.
Does that make sense?
Yeah.
But it will be carbon, hydrogen, and the side chain.
All right.
So hear me out. In nonpolar amino acids, you really don't have a lot of functional groups. The only one that I can legitimately identify is that thiol, well, thioether, but it's kind of an oddball. Other than that, it's carbons and hydrogens, carbons and hydrogens. Does that make sense? We don't count this nitrogen of the tryptophan because it kind of acts like carbon. When you start looking here, look at this. This is the ammonium group, but it's the part of that basic skeleton. You see what I'm saying? The part that is common. Is this an ammonium group? It is. It's just a functional group on the side chain. Side chains may have functional groups. Carbonyl, amino, hydroxyl. So, for instance, here, you may have a question. Why is this an amino acid and not this? Right?
Yeah.
Here's an explanation. This is ammonium, this is carboxylate. Agreed? How many carbons are, quote unquote, connecting them? 2. 1 and 2. Do you see that? That does not qualify as an amino acid in a biological sense. If you do this, then amino group and carboxyl group are quote-unquote connected by a single carbon. This is the must. Remember, naming, classification, it's an entirely human thing. Alicia, you have a question?
Yeah, so.
Go on.
When we're looking at them like this.
Sure.
The ammonium group and the carboxylate group are both in that blue, and that's our amino acid.
Right.
Okay, and then the extra stuff is
Side chain.
okay, because I was confused about the same thing. I was like,
Mm-hmm.
where did our carboxylate group go? But it's,
So let me do something here.
yeah.
So the CH2 would be the R group in this case.
What?
And like in the.
It's the part. It's all R group. Like this entire thing
Oh, that's, so that's.
is an R group.
All right.
Yeah, that's the.
Does that make sense?
Yes.
Okay. Now, we going into positively charged, it probably will like help to explain more. So hear me out. Look at lysine. Okay? What I circled here, this is the ammonium group. What I circle here is also an ammonium group. What I circle here is a carboxylate group. However, if you will say, well, I'm going to take this route, by definition, way too long. You see what I'm saying? This is the alpha carbon that connects amino and carboxylate. Does it make better sense now?
Yeah.
Okay, good. So these guys are positively charged. They're also known as basic, not in the folklore sense, but in the sense that the side chains can fairly easily accept hydrogens, like Bronsted. Lowry bases. Okay, and these are effectively not necessarily Bronsted Lowry, but those are acids. Okay, both Bronsted Lowry and Arrhenius. Okay. So I'm going to skip first a couple of bullet points. I think we got through this. You need to look for three-component backbone like this, okay? Okay, now you may notice I wrote it in a slightly different form. I will explain myself, like hydrogen here and only two hydrogens here. I will explain myself in a minute, okay? And then you look at the side chain, and that effectively determines the chemical properties of an amino acid. And based on the name, we use either a 3-letter or 1-letter abbreviation. Now, we're going to get to abbreviations right now, but so here's the thing. Traditionally, when we draw an amino acid, it is a convention that chemists accepted. I don't think it's even like formally written. But the good tradition is that the amino group is on the ammonium group is on the left, carboxylate group is on the right, and side chain, the R group, is either, depending on what you want, top or bottom. It doesn't really matter. Okay? Good? Now, naming. Overall, 3-letter acronyms are fairly logical. 1-letter acronyms aren't. So you may wonder why aspartic acid is D, and asparagine is N. Last letters. You can ask why not the first one, but the first one for both of them is A, okay? And there is also alanine, for example. So more than one. Anyway. So polar or non-polar? Glycine. Just a hydrogen on the side.
So non-polar.
Non-polar. And threonine. Remember the rule. The rule is what do you say? Polar, yes. The rule is to find something that is not carbon and then see if it gives an amino acid decent amount of polarity. Oxygen is not carbon, second most electronegative element in the periodic table, which means it's going to draw a hell of a electron density from the adjacent carbon. Good? Okay. Solution, we solved it. Oh my God. Valine. I need to go back. Like seriously, I don't remember the structures. I know that it's nonpolar. Let me get here. Valine, it's basically isopropyl. Okay. So I'm going to use this moment to illustrate something. So think about the name, amino acid, right? Let's put the central carbon. And we have an amino group. And we have acidic group. Now you see that I put hydrogen here and only two hydrogens here. I do it to tell you something, to illustrate something. This is an acid. Does everybody agree with me? It's acidic group. So acid, what can it do with a proton? Acids, do they give proton or do they accept it? Give. This is a base. What do bases do with protons? Accept. At physiological pH about 7.4, this hydrogen goes away. It is given away. Make sense? And this hydrogen is accepted by the amino group on the amino acid, making it charged also. Does that make sense to you? This is how an amino acid should look at biological pH. Now, structurally, valine, this. Okay? Cysteine, same story. But in this case, it's the polar amino acid that is cysteine. Okay? Well, yeah, naming. Usually first three letters. So, yeah, valine and cysteine, I forgot to do that. And since there are no other amino acids that start with V, and no other amino acids that start with C, from what I can remember, we have this. All good so far? Okay. So when amino acids interact with each other chemically, like they react with each other, they form a peptide bond. a bond between the carbon from the carboxylate group and the nitrogen from the ammonium group. Okay?
I just had a quick
Uh-huh.
question. As far as the naming of the ammonium group, like NH3, like isn't ammonia NH3 and ammonium is NH4?
Exactly. Let me explain. Good question. Hear me out. So, this is ammonia, right? When it accepts a proton, it becomes ammonia. Agreed? Normally, group like NH2 is amino. There is no specific term that would be different from ammonium if you add hydrogen to it. It's just the matter of convenience. Since the process of getting from neutral NH2 to positively charged NH3 involves the addition of hydrogen, it's the same process as this one. Am I making sense?
So it's all the ammonium.
Just ammonium, yeah, for convenience. Okay?
All right.
Good?
Yeah.
Okay. So the idea here is that when amino acids join together, they form that peptide bond, which has some very interesting properties, which I will briefly discuss. And the product of joining two or more amino acids is termed a peptide. Now, a peptide is several amino acids. Okay? If it's 2, dipeptide. If it's 3, tripeptide. 4, tetrapeptide. 5, pentapeptide, and so on, but usually we don't use that. So a peptide bond is basically an amide. So look at this. You have a carboxyl group of one amino acid, an ammonium group of another amino acid, and by losing water, it's a dehydration reaction. So two hydrogens and oxygen leave in the form of water, and now you have peptide bond. Okay, good? Now you may notice that we have a free carboxylate group on the right and the free ammonium group on the left. So we can just keep adding. Does that make sense to you? So what we've observed here is the formation of peptide bond between glycine on the left and alanine on the right. Okay? So when we look at the peptide, dipeptide like this, on one end you have the ammonium group. Do you agree with me? The end of a peptide that carries the ammonium group is called N terminus. The end of a peptide which carries carboxyl group is called C terminus. The naming starts at the N terminus. Okay? So the amino acid the first amino acid on the N-terminus is glycine. So we change glycine to glycel. And then the amino acid on C-terminus is alanine. We do not change it. Glycel, alanine. Okay? Now, if you don't mind, I really don't want to, I'm going to do something completely different because I'm very, very lazy. Okay? This is what I'm going to do. These are two amino acids, right? Serine and threonine. What I'm going to do, I'm going to do this and this. And then I'm going to draw a line, and that's your, well, technically speaking, hydrogen should be on the bottom, but it's fine. Okay, makes sense? That's your peptide. This is the peptide bond.
And it would have H2O left over.
Oh, yeah.
So is H2O always left over because, like, the peptide bonds basically are formed through dehydration synthesis? So it's you're taking water out of the molecules?
Mm-hmm. Exactly. Exactly because of that. So how do we name this? What do you think? Si. Okay. This is serine. This is threonine. Let me do the magic again. Nope, not serine. No, no, no. You replace INE with YL.
Yeah.
You see?
Yes. Several.
Yeah.
Yeah.
Yep. Okay. And let me work the magic of. Apparently it's gray. I don't know, whatever. I don't really care. So this is gone. This is gone. We're going to remove this, and we're going to add peptide bond and the hydrogen. Remember, again, it's a convention. If I want to put N-terminus on the right, sure. But people would think I'm a psychopath. So, N-terminus, C-terminus. All good? Let's move on. Well, yeah. What if you have more than 2 amino acids in the peptide? You start from N-terminus by changing the ending, yes, left to right, changing the ending from INE to YL. So, sorry, alanyl, right? Glycel. Imagine there are more, you keep changing until you hit C-terminus. The amino acid on the C-terminus does not change its name. So it is serine as it is. Okay? This. Alanyl. Lucille. So hold on.
E, right?
E is retained.
Oh, it is? And then that one says.
Yeah. So in some transcriptions, E is dropped or they do cystenyl. There is no real convention on that. I can guarantee you that. So it's, in my practice, it was always cysteal.
what would happen if there was like 20 amino acids joined together? Like, how long would that name be?
Not a single sane person does that type of naming
They would just shorten.
for long chains. It is done for 4-5 tops, because then you just use single letters. So this would be ALCM. Okay? So, well, we'll get to primary structure. Essential amino acids, these are essential for adults, which means we have to get them from the proteins. The only complete proteins, the ones that contain all 20 amino acids, are found in animal products. For vegetarians, there must be a combination between different types of vegetables. Okay? So now we're going to talk about protein structure, and there's something I want to chat while we're doing this. So, this protein is a polypeptide of 50. It is a fairly random number. It can be 49, and it's still going to be a protein. How do you judge? Okay? It's not a very well defined, like it's not, the protein with 50 amino acids doesn't differ that much from a protein with 49 amino acids. So it is an arbitrary threshold. Immediate question: dipeptide, tripeptide, tetrapeptide. What about 25? The usual term: oligopeptide. Oligo means a few. That's it. Have you heard about oligarchy? A power of the few. Okay? So when we use the term primary structure of a protein, we refer to the sequence of amino acids. Am I making sense? That's it. The order in which they are arranged. So, for instance, I'm not going to pause myself, but I'm going to say, I can't repeat enough. Stay in your lane. If you are a chemist, don't talk about endocrinology. Thyrotropin hormone that acts on the thyroid gland, resulting in the release of thyroxine, which is also known as, this is a thyroid hormone, okay, has this structure. So glutamic acid, polar, histidine, superpolar, proline, the only cyclical amino acid, and it's nonpolar. Okay? Why we pay so much attention to this? These 3 amino acids can be arranged in different order. Do you agree with me? There are several ways to arrange them, from left to right. I can put histidine first or proline first. Does that make sense? So there are ways. Only this sequence works as a hormone, thyrotropin. Okay, makes sense? So this is why when we refer to the primary structure, it is a fundamental feature of a protein. Like that what will eventually determine how it is shaped and everything. Here's another thing that I want to highlight. Remember we had a lab on the organic compounds, and you were constructing well, you were working with ball and stick models. You may remember that like carbon-carbon bonds, they rotate. Single bonds you can rotate around a single bond, right? So I'm going to ask you a tricky question. If you don't know the answer, you shouldn't know the answer, I think. So this bond, single or double, it is not a tricky question. Huh? Should the parts of the molecule rotate freely around it?
Yes.
Here's the tricky part. Listen. Oxygen has 2 lone pairs of electrons. Good? Understood? Nitrogen has 1 lone pair. So we have a lot of electrons here. We have a lot of electrons here. And we have a double bond here. So in reality, the peptide bond looks like this, where dotted line shows that electrons are 2 lone pairs of oxygen and 1 lone pair of nitrogen. They are distributed between the two, kind of spread. Okay, it's called delocalization. Not for the exam, but you are advanced enough to understand that. Which means this bond is a little bit double. Which means the rotation is severely limited. Which means there is a limited number of structures that a polypeptide can fold into. You understand? Like when you connect them, it's not they can turn next to each other on any angle. Go on, you have a question, I can see.
So, since they both have those pairs kind of near each other, it's like a brace between the C and the N bond. Say
again?
Do they like want to be on the same side as each other? Is that like why?
So these 3 atoms, carbon, nitrogen, and oxygen, energetically, it is better to spread the electrons over all 3 than have this uneven distribution. I'm sorry, I was a bit too... You see? Because that would be uneven distribution. Ignore these 2 dots, I can't be precise enough. Okay? So that's why they delocalized. It's always... A good friend of mine who is, he's a genius organic chemist, like seriously, and pretty famous in his field. He uses the word like, spreading the electrons. It's more beneficial. So what I'm trying to tell you is that if these are 2 amino acids connected by a peptide bond, it's not like they can roll around as they want. There's a limited angle they can turn relatively to each other. So there's some rigidity in that bond. Okay, make sense? So this is just an example of insulin, one of the smallest proteins that we know. So you see some people don't consider insulin a protein. They consider it a peptide because it is made out of 2 smaller peptides. You see? The A chain and the B chain. And these chains are connected by disulfide bonds. Those are sulfurs. Just sulfur, okay? We will discuss how they formed. I promise. We will talk about it. Okay? So we're going to answer, I am much better with recognizing an amino acid than drawing an amino acid. Like if you will tell me to draw the asparagine, I would have a bit of a trouble. I don't remember how many carbons in the side chain, but recognizing. So this, the amino acid on the left.
So would that be nonpolar then?
The one on the left?
Because
oh yes,
it's attached to that aromatic.
It's aromatic. There's nothing else in that side chain except carbons and hydrogens. So it's nonpolar. So this hexagonal structure is phenyl, okay? And that, specifically that, including that CH2, actually looks a lot like that amino acid. Do you agree?
When phenol for phenol, wouldn't that have to have that aromatic structure with like the OH with the hydroxyl group attached?
Nope. Okay, phenol.
Oh, I'm thinking.
It is, yes. Phenyl is this. By definition, this hexagonal aromatic ring, when it is attached to something, it is phenyl, like methyl, like ethyl. Okay. Now, this part and this part, do they kind of similar? So the one on the right is alanine, and the one on the left is basically alanine with a phenyl group attached. Phenylalanine. The one in the middle, there are only two amino acids that contain sulfur, cysteine, and methionine. Methionine is nonpolar, cysteine is polar. Okay? And the name-wise, phenylalanyl, cystyl alanine. Question?
Nope.
Okay. We good? Everybody? Cool. They use cysteine. I refuse to accept this. A lot of signaling molecules are polypeptides. So for instance, endorphins and enkephalins. So they are what we call natural opiates. Strictly speaking, okay, let's see the genesis of this concept. Hear me out. These are polypeptides, well, oligopeptides, okay? Like pentapeptides, okay? We're going to get to oxytocin. That make sense? Hundreds of years ago, people figured out that if you collect whatever the poppy seeds and do some magic with them, you're going to get something that makes you high. Later it was, and that substance was called an opium, okay? So when people figured out that there are receptors that opium acts upon, those receptors were called opiate receptors, okay? Later, when endorphins and enkephalins were discovered, turns out they act on the same receptors. So they were called opioids, although structurally, neither enkephalins nor endorphins have nothing in common. Like, they're not structurally the same as heroin, OxyContin, stuff like that. We just kind of bumped into a substance accidentally, event, many years ago. That makes sense? Okay. Another example of polypeptide is oxytocin and vasopressin. So oxytocin is known to regulate the labor through the positive feedback loop. It's also periodically implicated into emotional responses. And in one of my favorite studies in mice, it was shown that there is an oxytocin-mediated communication between the gut and the brain that probably plays a role in autistic spectrum behaviors, if it's dysregulated.
Autistic?
In mice, not in humans. But we can talk more about it. It's pretty exciting, but it's not chemistry, it's biology. I can say a few words. I looked up recently. So you see that oxytocin and vasopressin are similar in structure, right? It's a circular, it's a how many? 9 amino acids, 2 cysteines are connected by sulfide bond, right? And the difference is basically that in oxytocin, it's isoleucine, and vasopressin phenylalanine, and here leucine versus arginine. Well, yeah. Now, if you think about the function, oxytocin stimulates contractions of uterus and contractions of the smooth muscles around lactiferous ducts, producing the let-down effect, ejection of milk. Vasopressin controls the reabsorption of water in the kidneys and also regulates blood pressure. Okay? Vastly different functions, right?
So is vasopressin the same thing as
A-
a diuretic?
ADH, antidiuretic? Yeah.
Okay.
It's ADH, yes. So drastically different functions.
So would that vasopressin constrict your blood vessels?
Among other things, yes.
Because doesn't vaso mean blood vessels?
Blood vessels, and pressin refers to the pressure, yes. It increases pressure, yes. Now, hear me out. What do you think is going to have more effect on the structure of a peptide? The replacement of one nonpolar amino acid by another nonpolar, or replacement of nonpolar by a polar? The last one, right? When you change the polarity of an amino acid. Isoleucine and phenylalanine are both nonpolar amino acids. But leucine and arginine cannot be farther from each other. Leucine is nonpolar, arginine is polar charged. So I would bet that mostly this determines the target and the effects. Okay, names, we're going to say them. We're going to 3-letter, we're not going to do 3-letter. Glycine is G, alanine is A. Okay? So we have 2 glycines, 1 alanine. GGA, AGG. Oh, why it's different? Is that your question? They ask names and 3-letter abbreviations of the tripeptides. I just don't want to do 3-letter abbreviations. I want to do 1 letter. It's shorter. Does that make sense?
So if it was GGA, is that like glycylalanine?
Yes, because this is the N terminus, right? You see what I'm saying? And this is the C terminus.
Would you ever be able to put like diglycylalanine?
Yeah.
Oh, you can.
You can do usually we do glycyl because to avoid the confusion. Okay? Like, are they somehow connected in a different way? You can do this. And that's it. From what I can tell. Yeah. Okay? Now remember that in this case, it does matter. The direction matters. So GGA, glycylalanine, isn't the same as AGG because it's alanyl glycine. Okay? Does that make sense?